we will proceed to the mechanism of opening and closing of stomata. So this is a very important topic lah, for you guys. So I think this topic you also learn in SPM, but you are not learn in deeper, or maybe you totally not learn because you not like plant, because in SPM maybe you never come out yet, so you never learn about it, or you just screening out whatever you learn or not learn. But in STPM, if you want to score A, you should learn in detail all the topic that we cover in syllabus. Okay. Okay, so I think okay. So about the opening and closing of stomata. So normally, firstly, we will talk about the opening mechanism first. So if you see here, this is the general overall process of the opening stomata opening lah. So if you see here, the first process is normally stomata will open during the sunny day. It will open during the sunny day. So at night or windy day, when the light is not insufficient, the stomata will close. So why it like that? So why it only open in sunny day? So normally the sun in during sunny day, our light energy, if you still remember in your semester 1 that you learn in photosynthesis, you know that our light contain many color, right? Blue light, red light, green light, yellow light, all that, right? And then if you know that in photosynthesis, the two main color of the light is blue light and also red light, right? For the chlorophyll pigment to absorb. So similar to stomata also have the pigment that will absorb the light. So the light that absorb normally is blue light. Okay, so when the stomata is absorb blue light, the blue light is penetrate to the stomata, to the gut cells. Okay, the blue light will activate the proton pump, which is the hydrogen ion pump. Okay, hydrogen ion pump. So when the hydrogen ion pump, meaning that the hydrogen ion inside the gut cell will be pumped out, pump out from the gut cell into the surrounding lah, hydrogen ion. And then this one will form the electrochemical gradient. So if you still remember your chapter 3, the first semester, core transport process, you know that, right? And also ETC, transport chain. What is about the electrochemical gradient? It is one type of voltage, right? So now it already create the energy to create the one type of energy. And then once the hydrogen ion is already come out, another type of ion we call as potassium ion. Potassium ion will enter the gut cells. And then at the same time, chloride ion will also enter the gut cells. Okay, so once the potassium ion and chloride ion is enter the gut cell, the water potential inside the gut cell now is very low. Is very low because it's very low. So the water will enter the gut cell by osmosis, and then this one will cause the stomata to be open. So this is the general process. So now we go to, this one is also general process. Blue light activate proton pump, and then this proton pump will move hydrogen ion out from the gut cell, and then our potassium ion and chloride ion will diffuse into the gut cell through voltage activated ion channel, and then water will diffuse by osmosis into the gut cell, and then the gut cell will change the shape, and then stomata open. Okay, so this is more detail. So we go to one by one. So we know that the stomata open is during hot sunny day, right? Okay, when the blue light from the sun, so normally you know that blue light, the wavelength is between 400 nanometer to 500 nanometer, will trigger the activation of proton pump. So I already mentioned when it trigger proton pump, so the hydrogen ion inside the gut cell will actively transport pump to outside of the gut cell. So this one will create the electrochemical gradient. So this one is one type of core transport. Okay, core transport. So forming there are two sides of the gut cell plasma membrane, meaning there are charges between the there are the concentration difference between the gut cell plasma membrane. So this resulting electrochemical gradient will drive the facilitated diffusion of large number of potassium ion into gut cell. So when the hydrogen ion is pumped out, you need to balance the charge, right? You need to balance the charge. You not say that when the hydrogen ion, meaning that the hydrogen ion is pumped out, meaning that the charge now in the gut cell is imbalanced. Is imbalanced. The gut cell now is highly negatively charged. So you need to balance the charge. So another positively ion, what you call as potassium ion, will pump into it. Through not pump, through the facilitated diffusion through the potassium ion channel. Okay. Okay. At the same time, also chloride ion will also diffuse into the gut cell also to maintain the electrical balance caused by the positively charged potassium ion. Yeah, although potassium ion is positively charged, but we know that potassium ion is highly reactive lah compared to hydrogen ion, right? You learn in chemistry. You learn in chemistry, you know that potassium ion is highly reactive compared to hydrogen ion. So you need to balance it again. You need to have a chloride ion also. So the chloride ion at the same time also will diffuse into the gut cell. So not only potassium ion, chloride ion will also diffuse into the gut cell. So when the potassium and chloride ion accumulate in the vacuum of the gut cell, this one will increasing the solute concentration in the vacuum. So that's why I say that when the potassium and chloride ion is enter into the gut cell, the water potential of the gut cell now is very low. Okay, so if you know that water is moved up, the movement of water is through osmosis. We know that osmosis process is the process of movement of water from high down the concentration gradient, right? From a high water potential to low water potential. So now the water will enter the gut cell from the surrounding epidermal cell. We know the gut cell, the surrounding of the gut cell is epidermis cell. So all the water surrounding the gut cell will enter into the gut cell by osmosis. So this one will increase the turgidity of the gut cell and change their shape. And then the thicker inner cell wall will not expand as much as the outer cell wall. So this is the reason why the inner cell wall must be thicker compared to outer cell wall. Because when water is entered into the cell, into the plant cell, we know that when there are many water is pumped into it diffuse into the cell into the plant cell will become turgid, right? Turgid. So when it is turgid, the plant need to expand it. The plant, it will change the shape by expand it become like this. So only the outer cell wall of the stomata is expand only, but the inner part is not expand. It maintain, normally is maintained one. The expand part is outer one. So you will see here, this one is closed one. So closed is like this. But when it is opening, the outer part, the curve shape is more highly curved compared to inner part. So that is why that your inner cell wall must be very thick. To prevent the inner cell wall also will become more curved like that lah. So this is one of the reason lah. Okay, so that's all for the opening stomata. So once like that, the stomata will be open. So this is all the mechanism about the stomata opening. So it is very easy. So this is the process. You just remember this process. In doing essay question, just write out this process only. The most important thing is the blue light. Blue light, not only sunlight. In exam, if you write sunlight wrong, not accurate, you need to write blue light. And then proton pump, I think this one not problem. And then you need to say proton pump is out of the gut cell, not proton ion, not hydrogen ion in masuk ke dalam gut cell, kan? Dia keluar daripada gut cell. Okay, at the same time, potassium ion and chloride ion, these both ion will be diffused into the gut cell. And then through what? Through voltage activated ion channel. It has the protein channel, spatial protein channels, unique for only potassium ion and chloride ion respectively. And then this process is called facilitated diffusion, not simple diffusion, not active transport. And then this one bottom part is active transport. This one is facilitated diffusion. So all the process you need to mention it. Although this one I think you learn in semester one, right? So what you learn in semester one, I did mention in sem one, right? If you attend my class, all that you learn in sem one, you also need to apply in sem two also. So you cannot forget it lah. So for those who already forget, please keep it. Keep it what is this process meaning. And then after that, once the potassium ion and chloride ion diffuse into it, due to the accumulation of these two ions will decrease the water potential. So the water will defeat the water from the surrounding stomach gut cell will diffuse into the gut cell by osmosis. So once the water is all many water is enter the gut cell will become turgid and change its shape and cause the stomata to open. So that's all for the mechanism about the stomata opening. So I think very easy, not very hard. Okay, now we will proceed to closing. So closing is totally, you can say, is reversible reaction. So normally in closing, normally is at night. Okay, during night, there is no light at all, no sunlight already, no blue light at all, right? Because what if you want to make a stomata open, you need to have a blue light first. If no blue light, the initial energy, then stomata will not open. So if you want to close the stomata, then you just cut down the blue light. So once there are no blue light at all, the water molecule inside the bottom part will be inactivated, and hence your active transport will not occur. Meaning the hydrogen ion is maintained at your inside your cells. So no active transport, meaning that your concentration of potassium ion will be slowly decreased. It will start the potassium ion inside, because the potassium ion is not the natural ion that should be maintained inside the gut cell one. The natural ion that should be maintained inside the gut cell is hydrogen ion. So now active transport is not occur, meaning that the hydrogen ion at the surrounding here now will start to diffuse back into the gut cell by facilitated diffusion. So once the hydrogen ion is starting to diffuse back, your potassium ion and the chloride ion will start to decrease lah. Okay, so once your potassium ion and chloride ion start to decrease, meaning that your water potential inside the cell now is increased. Now is increased already, because all the solute, because potassium is one type of organic, one type of nutrient, right? One type of mineral ion also. So once your mineral ion, mineral salt, all this already lift out, meaning that your solute potential now is decreased and your water potential is increased. So once your water potential is increased higher than the water potential that in the around the gut cell and the epidermal cell, your water will start to diffuse out. Okay. So when your water is start to diffuse out, your gut cell will lose their turgidity. So you know that when in plant cell, when the water is diffused, when the water is lost from the cell, the plant cell will become flaccid, right? But this in this stomata condition will not become flaccid at all, like the that you learn in SPM hypertonic condition. No, it just will maintain until the equilibrium. Then the water will stop. The diffusion of the water will be stopped. And then once the water is diffused out from the gut cell to the surrounding, the stomata will lose the turgidity, and then the turgidity meaning that the cell wall now cannot be expand, or cannot maintain its curve shape. Dia akan macam ni lah. So this one will cause the stomata close. So it will become like that. So it will become origin. So this is the very simple. You just know that when you want stomata open, you need blue light. So when you want to curse the stomata, then you cut down the blue light. When you cut down the blue light, then no anti-transport, and then confirm your potassium ion and chloride ion will start to diffuse out, and once the potassium ion and chloride ion diffuse out, your water will be increased, water inside the gut cell increase, and then this one will also cause the water diffuse out from the gut cell. And then once the water diffuse out from the gut cell, the gut cell will lose their rigidity, but it will not become flaccid. So you cannot write the gut cell become flaccid. No, the gut cell will not become flaccid. It just lose their rigidity, become normal. And then once it lose their rigidity, the stomata will be closed like this. Okay, so closing is more simple compared to opening. So, okay, that's all for today. So today's topic class is very short. So I hope it is this. So I open Q&A for now. So any topic you want to ask, not only plant, any maybe animal that you not understand, you can ask also. Afterward, I will call, and starting next week, I will start new chapter lah, chapter 8, transport lah. So chapter 8 is more quite longer chapter compared to this chapter. So maybe I will cover more topic lah in one day because your semester 2 is very short. So I need to move faster lah. Okay. Okay, I think that's all for today. So any question or not? I give you maybe few minutes until 8:26, 27 like that. Let you type your questions. Mm. So when you type, just talk something lah. So in overall, this chapter is quite easy chapter, not very hard. I can say it is the easiest chapter lah. One of the easy chapter for this semester because start from next chapter it will quite hard lah. But chapter 8 not hard not hard lah, not hard, very hard, moderate lah. But when it comes to hard is starting chapter 3. Starting chapter 3, many new things, and chapter 4, chapter 5 lah. So yeah, so you need to be alert lah because now is still you are third week, right? Third week that you enter your semester 2. So yeah, maybe you are in honeymoon yet. I don't know. So I don't know. School teacher already did it finish up one chapter or not. But should be lah. Should be finished up lah. No question. Got question? No question? Okay, so I hope that everyone understand my class. So if no question, so I will end the class today. So today class is very short. So okay, so next week I will enter chapter 8, transport. So okay, so I think that's all for today. So thank you for your attending and good evening and good night. Bye.